Good morning, everybody. I am Kristin Smedley, one of the co-founders and now CEO of the CRB1 group, patient group, global patient group at the Curing Retinal Blindness Foundation. It is my incredible honor that all of you are tuning in either live today or watching this later to be a part of what I've been calling the magnificent mission that I cannot stop smiling about, because of the folks that you're going to hear from today. And there's folks in the background room, the attendee rooms, that have been a part of this since we started. And I could not be prouder and more honored to lead such an incredible team, an unbelievable mission that I joke when I present at rare disease conferences around the world that I'm so glad I had no idea what I was getting into because it is way bigger and broader than I could ever have imagined. And it truly is magnificent. So, it's actually nearly 22 years ago that my journey with CRB1 blindness started. And honestly, it was right after when everyone was trying to figure out if at Y2K, if computers were going to crash and if the world was still going to be here. It turned out that we were still here on January 1st of the year 2000. And 29 days later, the most miraculous thing happened. I became a mom, and I had dreamt about being a mom for a very long time. And for 4 months, I looked at my little Michael and envisioned pitcher on a baseball team and quarterback of the football team, valedictorian, all the things that you put all those hopes and dreams on that little face, right? And I was right there with all the other new moms doing the same thing. And then at 4 months old, a doctor said to me, Michael's blind, and we believe that it's LCA. And as those of you that are watching and here with us, if you've heard those words, then you know what a sucker punch that is to your heart and to your hopes, because the second sentence that was the worst thing that anyone could ever say to me was, there's no hope. So that was back in the year 2000. There was absolutely no hope. I remember the doctor had said, maybe, Chris, I said, how could there be no hope? And he said, maybe in 50 years, there might be a glimmer of hope, but right now there's nothing. So it would take me a while, and you're going to meet some people today that helped us turn from hopeless to hope fueled, because it would take me a while to get my head around raising a blind child. And 3 years later, hearing the exact same sentence, your son is blind, with my second son, and hearing again that there was no hope. But, I went looking for hope, you guys. I looked and looked for hope, and I found a little crack in the door here with an organization that my good friend Irene Maumenee is going to be talking in a few minutes, that she introduced me to. That helped me figure out how to raise my boys to thrive. And then there was a crack in the door of hope when I saw a news broadcast about a team here in Philly that was making some progress for a different gene similar to ours. Cracks in the doors of hope I was looking for everywhere. And then I finally started finding some good hope when that RPE65 trial went underway. And then it was time to fund hope. So I found some other parents through the power of Facebook that were interested to find and fund hope as well. And that's what we've done for the past 10, almost 11 years here. Back in 2011, we got together, just a very small group of families that were interested in funding hope. And boy, have we funded hope. Like I said in the beginning, if I would have known what it was going to take to get this far, it's just been absolutely extraordinary. We've been involved in the Luxturna FDA approval. We went and were a voice to make that happen. We've been on the floor of Congress. We've got a patient registry underway. We have cohorts all over the world. Thank you to the patient leaders from around the world that are in the room with us today. We are finding the patients, empowering the patients and their families, and best of all, connecting with experts that are dedicated to seeing this thing to the finish line. And you'll meet just a few of those folks today that we're honored to have with us today. But, we've done all of this funding hope. But now we have our eye on the finish line. And you're going to hear about some new things that are happening in the world. You may have already heard some of the words that you're going to hear today, some of the technologies, some of the breakthroughs, and we have an opportunity to fund that finish line that we have our eye on. We're going to talk more about that in a little while. Right now, I want to, as I said, introduce you to a good friend, Irene Maumenee, that, you know, years ago, I went to her with a tiny baby in my arms and said, "You please help me." And she directed me to those incredible resources to help my boys to thrive. And then I went to her when it was time to, when we found hope and we wanted to fund it, and asked her if she would help. And she said, absolutely yes. And she's been with us this entire journey. So let me switch this over to Irene. And I'm gonna take myself off of the screen and put Irene on it. Irene's gonna give you a little heads up on what CRB1 is, where we're at, and where we're going. Hi, Irene.
Okay, hello. Okay. Your video, okay, that's great. Good. Hello, everybody. Thanks for coming. Thanks for joining us. So this is a short update of where we were, are now, and are going to go. And the remaining questions, and what will we do with the next 10 years? And will we, in 10 years from now, what will we see and where will we be? So I'm glad, and I hope I get all the input from everybody. And here I am to share my screen. Okay, let's put it on, come on, I've done that more than once. Where is it? There it is. Okay. Okay. So I'm now at Columbia University. I spent about 4 decades at Hopkins and close to a decade in Chicago. And when you add up the years, that certainly adds up. And I hope that I'm still here in 10 years, what we are aiming for at this moment. So what happened in finding the gene for CRM1? First, Bleka Wagemakers and her friends out of Amsterdam described a Dutch isolate in which there was sort of a rare eye disease, and they found quite early that basically there was a lot of variability in the clinical phenotype between the patients. And then Anneke van der Hollander and Francis Kremmer's group in general went and found the gene and published on it in 1999 and 2001, and they named it after Drosophila model that where the gene had been known. Then genotype-phenotype correlation studies started, and there was they remained sort of inconclusive. Juliana Salom from Brazil, who may be here today, found some correlation in her patient data and other people did not. So this is still up for debate. And then Kristin and her family and friends then started the CRM1 Foundation in 2011. That is now 10 years ago. Then the phenotype was expanded by adding a central type to the already known types, and we see that in just a minute. And then more recently, Jeremy Kay described other isoforms of the gene in contrast to the standard form, and expanded the field dramatically in the year 2020. And then people have been working on organoids, Marius Oeffing and Pete Quinn primarily, and we will hear about that. Natural history studies in order to see what would be a good parameters for measurements once clinical trials start have been ongoing, and there are some positive results. CRB1 is the second most commonly involved gene in LCA after CEP290. And as you know, CEP290 is undergoing clinical trials now. So we want to get CRB1 there. It is also the fourth most commonly involved gene in RP. The numbers are really vary by country. There are a lot of patients with CRB1 LCA in the Middle East. And so to really come up with a good estimate is somewhat difficult. The frequency of LCA varies. The estimates vary from 1 in 30,000 to 1 in 200,000. And if you think it's 1 in 100,000, I think that's a reasonable number. And then you think that CRB1 is the second most common. So, you know, where do you finish up? Maybe 1 in 200,000. But other people counting numbers will come up with estimates of 1 in 80,000. So it's a little bit up in the air. The characteristic fundus findings consist of sparing of paraarteriolar RPE, which is what John Heckenlively described many years ago. Then there are nummular pigmentations, which are quite typical. And in a large proportion of patients, you see as time goes by that they develop a coats-like response, which is that they develop vascular changes in the retina, which are sort of the terminal changes in the eyes. And Juliana always says it's just a question of time. As you wait long enough, all patients will get that. The youngest I have seen that is actually in a teenager, but I think it's more a late development. The gene is located at chromosome 1 on the long arm, and it is needed, the protein is needed for both outer limiting membrane integrity and photoreceptor morphogenesis. Mutations in CRB1 lead to 4 partially overlapping phenotypes. There's Leber's congenital amaurosis, early onset retinal dystrophy, and early onset retinal dystrophy and Leber's congenital amaurosis, they certainly overlap. So you define them whether somebody gets nystagmus or not, but really they're very much in one group. And retinitis pigmentosa, which is severe phenotype, and then patients have a macular dystrophy. These are all autosomal recessive conditions. Is there a late manifestation in the heterozygous state? We don't know that. And there haven't been enough studies on that. We had that in our earlier questionnaires, whether there are more patients with AMD or related disorders in the grandparental generation, but we don't know the answers. Here's a fundus picture of this. And when you look at these pictures, and it takes a little more of a study, you can actually see that within a year, there is a significant change in the central lesion that is seen in this patient, in these patients. Basically, looking at the natural history, logma, visual acuities, foveal thickness, and size of the central lesion may be useful clinical variables for measurements of treatment effect. So I think we are there where we can have parameters that we can measure for the a treatment outcome. What we are now heading for and what we really need to figure out is what are the ideal treatment modalities? What are model systems? Do we really need model systems? Besides the organoids, what is necessary? How do we organize clinical trials and what's happening there? We want to get to FDA approval. What's the timeline for all of that and what is the cost? We will head back and hear more about all of these points and maybe some others that I didn't think about. And we now have Dr. Fyodor Onov from Berkeley, who is going to talk to us about his work on CRISPR and related modalities. Fyodor, your turn. stop share. We are not going to put questions in there. We'll go through questions as we go along, unless there's some very urgent one, while we load up Fyoda's talk.
Okay.
Kristin, you are.
Thank you for the gracious introduction.
It was short.
It was perfect.
Okay.
I work with Jennifer Doudna in the building that you see right behind me on the image. Our life's work is to build and put into the clinic CRISPR-based therapies for precisely the sorts of genetic conditions that LCA CRB1 represents. And when I got the invitation to speak with you today, I deliberately put a talk that is first 4 minutes and 30 seconds long, two, extremely aggressive in its claims. And if there's one thing I want the CRB1 community and my colleagues to take away from what I'm about to say is we, the editors, are ready to go to the clinic for CRB1. And this is not one of those, oh, but here are the 55 unknowns. We know what they are and we know how to de-risk them. It is a matter of coalescing the team and going forward. And in 4 minutes, I've used up 30 seconds of my time. I will tell you, based on our current experience in Berkeley, California, that this is start to finish, 4 years and $6 million academic pricing to actually dose a human subject at UCSF or Penn with a CRISPR for the common allele of CRB1. And how can I be so ambitious? Because of data. I'm a scientist, I'm a molecular geneticist, I'm a gene editor. So let me tell you in now 3 minutes, 45 seconds. People accuse me of speaking too fast. Well, I'm going to set my own land speed record here. So just real quick, I want to make sure that the isoform of my screen that you can see, can you see the full side display or the speaker one?
Presentation view.
Okay. How about now?
This
Yeah,
is
it's good.
All right, folks,
Yep.
the time is now. In 3 minutes, I will show you this image and explain why this is the therapeutic that you just need 4 years and all that money to put into the clinic with. Bear with me, in 3 minutes, this will be clear. I worked with Jennifer in 2012. She did a Nobel Prize winning study with Emmanuel Charpentier in which she proposed that CRISPR-Cas, that's in the news all the time, can be used as a medicine. Remarkably, that has been proven true. Editing did not originate with Jennifer's work, but progress was extremely slow in the clinic from 2009 to 2019. The reason I'm so ambitious but data-based, and my promise to you that it's about a 4-year timeline to get to first in human, is this is what's happened in terms of promising clinical data. This. So the original isoform, which is not, I think, what you should use for CRB1, but I think is important for you to be aware of, is creating a double-strand break with this programmable enzyme, which I won't review because many of you know it. And all of this is in the clinic, as I'm about to describe. And that paves an important path for what the CRB1 community could consider doing, including, as I'm about to describe, the newest data from CRISPR editing for LCA. But let me start by pointing this out. There is nothing other than effort and money standing between the CRB1 community and this gallery of edited people for 3 different diseases from having a 4th person. And Jennifer and I speak all the time about the fact that the worst thing we can do is give people false hope. We work with rare disease foundations all the time. We never give false hope. What I'm saying is actually realistic. And the reason it is realistic is because of the courage of the people you see before you who have been edited for their sickle cell disease, TTR amyloidosis, and LCA. And together, that clinical and preclinical effort has given the Food and Drug Administration, the EMA in Europe, comfort to allow in vivo gene editing. Specifically, we have 27 people who have been CRISPR cured of their sickle cell disease, 6 people who have been treated with good efficacy with editing in vivo for their nerve and heart disease, and perhaps most importantly, 6 people who have been gene edited by eye injection of CRISPR for their blindness. And I'm going to speak briefly about what those data are because my time is short, but if you'd like a 3-hour presentation on what that looks like, happy to coordinate. So I really salute Editas Medicine for taking on LCA as a very challenging indication. And over a 6-year effort, a good fraction of which is public and thus provides takeaways for the CRB1 community, to do the cell studies, mouse studies, monkey studies, manufacture, regulatory, and actually dosing 6 subjects. These are data provided to me by a good professional colleague, Lisa Michaels, who's the Chief Medical Officer of Editas Medicine. And data on one of the subjects shows trends towards improvement. But the single most important point the CRB1 community should take away from this is not the efficacy signals. This is going to take them another 4 years to do, but that the Food and Drug Administration allowed them, based on the safety and preliminary efficacy data, to advance to dosing a cohort of pediatric subjects. And the agency would never do that if they saw safety signatures or lack of efficacy signatures that would give them concern. So this is a tremendous step forward, not just for CRISPR, not just for LCA, but also specifically for the CRB1 effort. So key thing, I'm not going to spend any more than 10 seconds speaking about this. Going to the clinic for CRB1 should not use the classical editing currently in the clinic. It should use the next generation genome editing, such as base or prime. These tools exist, they are available, and they can be engineered. And in fact, the middle one just went to the clinic. Again, another subunit of the overall problem space solved. Wrapping up, Luxturna showcases the feasibility of getting an approved medicine for an eye disease. Experience with Millicent, which is antisense therapeutic engineered by Tim Yu at Children's Boston One Child, is enabling from a regulatory perspective. As I just told you, CRISPR is in the clinic for multiple disease indications, and this means there is a fully charted regulatory path from disease to genetic target to first in human. Editas clinical trial for LCA showcases the feasibility of getting to the clinic using in vivo editing. There is no such thing anymore as a mutation we cannot repair. Peter graciously sent me the dominant allele, and it took me 30 seconds to pull up this image in the book of human DNA. This is the human genome browser, to identify that specific mutation. And this thing in green, or this thing in green, is the string of nucleotides that we should be targeting today using CRISPR-Cas9. We have never had a technology where the first principle design of a therapeutic takes a PhD in molecular biology 30 seconds, but the big work remains. The example of Kathy High and Luxturna and Penn is exactly the right one to quote. The academic nonprofit setting is well suited for efforts of this type. I'm wrapping up. Getting in an academic nonprofit setting, such as California or Penn, in any in vivo clinical trial using viral delivery is 4 years start to finish. In academic medicine today, it's about $6 million. These are CERM pricing, California Institute for Regenerative Medicine, how much it costs to get to IND. This does not include clinical trial costs. But critically, and I'm closing up, the bits and pieces are there. We only have Rumsfeldian known unknowns. They just need to be coalesced into a conjunct, vertically integrated effort. And for that reason, for driving this effort is what I salute Kristin and the families for, because they exemplify our central operating principle, which of course is the principle of all parents and families of children with such diseases. And it comes from the United States Marine Corps. Quote, courage is not going into a room full of fear unafraid. It is being afraid and still going in. And my last message to you is, we now have a rational, strong basis for going into the room of putting CRISPR in the clinic for LCA CRB1. Thank you.
Wow.
Wow.
Thank you for that. I'm wondering if maybe you have some Philly roots, because you talk as fast as we do here in Philly. That was great.
I think we'll have you come for 3 hours in the near future and join us more often. So this is exciting. I like your timeline, but I'm including the clinical trials into the FDA approval and all of their time. So in 10 years from now, we may be doing a good job. So here we go. Now it's Pete's turn. And you all know Pete Quinn, who's now at Columbia, and he will talk about prime editing of CRB1 associated inherited retinal diseases. Pete, here you go. Behind him is Steve Tseng, who is running the DNA lab at Columbia.
Yeah, so hi everyone, nice to be here. What I do at Steve's lab is investigate inherited retinal dystrophies. And actually I've been working in CRB1 for more than 10 years because I did my doctoral studies on it as well, making animal models and organoids and also gene therapy. And I'll just talk you through a little bit of the things that we've done and are going to do in the future. So yeah, as Irene pointed out, Chromosome leads to a spectrum of different inherited retinal dystrophies, including retinitis pigmentosa and LCA, which are much more severe than some of the others. And there's a lot of chromosome mutations. Actually, there's now even over 400, and they're placed throughout the gene. But there's still no clear genotype-phenotype correlation, and we know that might be true now because there's multiple isoforms that I'll talk about in a moment. But canonically, the role of this protein is for stability of your different cell types, so for tissue integrity, and it's for the adhesion between one of your main light-sensitive cells, your photoreceptors. And of course, if this adhesion breaks down, then you can start getting degeneration and blindness. And there's nuance species-specific differences, which makes the kind of modeling that Irene was talking about sometimes difficult. And that's why, for instance, myself and Marius also are moving to these kind of human mini retina organoids to try and model this disease. So last year, actually, Jeremy Kay from Duke found a new isoform of Chromosome, so this Chromosome B here at the bottom, and you can see it's much shorter, but it shares a lot of homology, so similar sequence to the canonical Chromosome A. And by itself, it could be an interesting therapy, a gene augmentation therapy. But what importantly he found is that Chromosome A, so the canonical one, and Chromosome B, the new isoform, are in two different cell types. So they're in opposing cell types. And we did an analysis of the different chromosome mutations. And what we found is that about 70% of mutations affect A and B together. So not only are A and B in different cell types, but most mutations affect A and B. So this kind of makes the therapeutic strategy a little bit more difficult, and maybe makes kind of classical gene augmentation a little bit harder than we thought. So just to quickly go into that. So gene augmentation is where you supplement a healthy copy of a gene into a cell in the hopes that it can restore function whilst you keep the defective one still there. And actually, Jan Weinholz, my previous PhD mentor, has some good proof of concept work of this for Crohn's that's still in development. But what we're going to talk about here is gene editing. So that's where you use different editors like we talked about previously, and you try and repair the DNA that was damaged in the cell to restore cell function. So most mutations are kind of simple point mutations. So that's just simple letter swaps from a C to a T. And these simple letter swaps, actually we experience these every day and normally are fine, and we can repair these or that don't cause so much problems. But if we experience these in early development, or if we inherit these from our parents, this can cause a genetic disease. And as an example, so here's a patient in the black square who inherited 2 mutations from his mother and his father. And you can see that on the right-hand side, there's a series of peaks. And you can see where the arrows are, there's 2 peaks. And this kind of represents the discrepancy between the healthy sequence from one parent and the unhealthy sequence from the other parent. And this is what we're trying to repair. So as we said before, there's kind of 3 different flavors of gene editing. So there's kind of nucleases, which have been considered these kind of molecular scissors that can come in and cut your DNA. There's base editors, which are kind of like a pencil, so it can change one of these base pairs to another. And there's kind of prime editing, which is the search and replace gene editing, which is more what I will be talking about today. But what I want to tell you is that depending on the type of mutation, any of these options might be interesting to you. So we have to kind of develop therapeutic options sometimes on a per mutation basis, and this needs to be carefully considered. So I won't go into all of the details about prime editing, and maybe at another time, like I did last year, I can go into a lot of detail about the different types of editing. But prime editing allows us to kind of fix these point mutations, so these single letter swaps, these small insertions where you have an additional letter, or these small deletions where you've lost a couple of letters, and we can put those back in with prime editing. So how does prime editing work? So it uses this kind of Cas protein, which is a bacterial defense protein. And if we give a guide, use a guide RNA, this can kind of target to the right place in the genome where we want to edit. And then prime editing, there's this thing called, it's being fused to something called reverse transcriptase, which allows us to use an RNA template to start making DNA. So we can start incorporating a repair template to fix the mutation where we had the problem. So can prime editing treat chromosomal mutations? Well, we did an analysis of this, and actually, based on about 300 mutations, prime editing could nearly theoretically fix all of these. So it's a great potential approach for editing. And actually, we also found that the top 10 list of mutations that were most represented in patients, and the 948 one, as was shown previously, and actually in a number of papers is shown to be the most common one. And this would be an obvious target to go for. So this is something that we've done. So here's an example of 2 different patients with this mutation. And then we use prime editing to fix this. So so on the left-hand side, you see this discrepancy again between the parent and the 2 different parents. So one mutation is bad, and we fix this using prime editing in these stem cells. And as another example, so this is another chromosome mutation that we've been working on. And again, using this prime editing technology, we can repair this in stem cells. So now we want to move this approach to kind of a more representative cell type. So one way of doing that is using these retinal organoid models. So these organoid models, as Marius will also probably talk a little bit more about, kind of represent the retina that we have in our own retina, and they have all the different cell types and cell layers. And we can use these now to model Chromosome retinal dystrophy. So this is actually work that I did during my PhD, and we managed to find reproducible phenotypes in 3 different retinal organoid models from Chromosome retinitis pigmentosa patients. And now also here at Columbia, I've also been doing this with Chromosome LCA. And what I want to highlight with this is that we can now find measurable outcome measures. So these are things that we can then look to see that are ameliorated or treated when we provide a therapy. So kind of the future of my work is to bring these 2 things together. So to bring the prime editing to these kind of post-mitotic, so these adult retinal cell types, and see if we can then fix these organoids. So just as a kind of summary, so we have chromosome A and B, and they're in these different cell types. Most mutations affect chromosome A and B. Prime editing or base editing are very viable options for fixing these, and we need to evaluate this on a per mutation basis. Yeah, we've had very good efficiency so far using prime editing to correct chromosome mutations, and we actually have a recent provisional patent out on this. And yeah, using this technology with these retinal organoids, we hope in the future to show that we can fix this this disease in these models. Thank you for listening. And yeah, this is the team that I'd like to acknowledge.
Great. Thanks, Pete. Again, maybe we'll have one question fast while we upload Marius Uffing's talk. Marius is, you want to look in the question if you have something you want to do.
May I ask a very short question? This is Fyodor. May I ask a very short question?
Yeah, sure.
Pete, just to clarify, congratulations, you're doing all the right things, just from an editor's perspective. You mentioned developing delivery methods with organoids. Any reason just to not do AAV? I mean, Editas has sold that.
So of course there's the dual delivery that you can do with prime editors now that's been published. So that's one AAV method that we could try. There's packaging limitation of prime editors, right, is the problem. So actually at the moment we're working on potential AAV and also lentiviral methods to deliver the prime editors.
Great. But the good news is, just for the entire community, if you show up in front of the FDA today with a dual AAV using a prime editor for eye injection, they'll say, they've seen it. So a tremendous advantage from a regulatory perspective and path to the clinic perspective is you don't want to surprise the agency. And so if you show up with something they've seen before, their level of comfort with reviewing your package is going to be in a different space. So I think delivery innovation is 400% what to do. Okay. And I really salute you for that, because ultimately viruses are ridiculously expensive. AAV is $2 million. GMP grade out the door, 2 million, just one batch. So it's not the way forward ultimately. But in terms of getting to first in human, what the agency knows is an incredibly powerful thing. Congratulations. I really was thrilled to see your talk.
Thank you.
Okay, good. Can we get Marius to talk?
Can you see me? Can you see my screen?
We see it. You want to enlarge it. We see it.
Yes. So better now?
Perfect. Perfect.
Okay, so thank you for giving me the chance to show what we are doing here in Tübingen. So the first thing is to actually inform you about what we think about treatment development. So before talking about treatment, we must understand the actually background and the molecular mechanisms of disease, and then we will determine which therapy might be or might not be successful. So that's important. Without understanding, I think treatment is very difficult. So this is this concept of systems pharmacology, where you compile and generate content-rich data, clinical data, omics data, functional data, and similar to what Pete has just alluded to, it's important to understand mechanisms and have the models to understand the mechanism. So then you are able to generate a hypothesis and validate that. And it's very important also to falsify it and fail early if your hypothesis is wrong. If it's right, you target, model, and validate the right pathway. And what I'm talking about is mutation-independent therapy development. Select the right target, select the right model system to study the disease. It could be human organoids, it could be a mouse model. I think in the case of CRB1, the human models are much better than the mouse models, because the mouse has the different use of the different isoforms of CRB1, and then generate a preclinical proof of concept and plan and go for a clinical trial. And here I think safety comes first, but also treatment modalities matter a lot. And at the end of the day, you have to define the right patient cohort. We have seen that in the 3 investigator-driven gene therapy trials, which we have been launching in Tübingen. It's very important to have the patients carefully selected. So what we are seeing here is quite a number of patients that suffer from CRB1 mutations. And I want to present you 2 brothers. They're both RP patients in our ambulance, and they differ with disease severity, although they are not far away with age, 25, 23. One of these brothers has a very severe form of RP. He's blind. The other one, a less severe form, and he can see quite well still, especially in his central vision. And of course, if you set up a systematic approach, you need the healthy control. So we have been actually creating organoids where we have the mutants and the isogenic controls. And at the same time, we have been actually doing a systematic interactome screen, which is to find out which the protein partners are that CRB1 is using for its function. The main goal here is not to investigate primarily what makes these 2 brothers ill, but to investigate what gives the one brother a chance to still see, to be less severely affected, whereas the other is blind, and to find the protective mechanisms that saves the one from getting blind early. And we do that by multi-omics, looking at gene expression, RNA-seq from the organoids, and we go into in-depth analysis of the organoids from these 2 brothers in comparison to the control. So these 2 brothers have different disease phenotypes. We have the organoids, and what we see, and this is really exciting us, if we have a control, that control organoid is light responsive, and if you flash light on the organoid, you get a light stimulus evoked transmission of the signal, and you get ganglion cell activity, which we can measure on a multi-electrode array. This doesn't happen in patient A, who is severely affected, but it happens in patient B-derived organoids, which tells us that this modifier must be actually inborn. It's in there. It's not based on the age difference. It's based on genetics. And so the disease modifiers could eventually serve as therapeutic target for CRB-linked retinal diseases. And it could be actually either an adjunct therapy to gene therapy or CRISPR-based therapy, or a therapeutic approach on its own. So as I said, we do a gene expression analysis from both brothers, but we also sort the cells from these two individuals, from organoids. And for example, we look at the Müller cells and the Müller cell behavior separately using a couple of different high content screenings, for example, calcium imaging, proliferation, and also pathway analysis currently focusing on the Wnt pathway. The second approach is to look carefully on the interactome of the CRB isoforms. And the goal here is to get a retina-specific interactome, because the blindness is, I think, one of the main features which we want to address. It's the key feature of this disease. And so we look into the interactome using porcine retina as a lysate, and we fish protein interactors in a kind of interactome screen by mass spectrometry to learn what CRB is interacting with, and then integrate these data on a multi-omics way. And so the approach is very simple. Show me your friends and I tell you who you are. So if we know the interactions that CRB will take to exert its function, we learn about how CRB will function as a kind of normal, healthy protein and as the mutation-affected proteins. And so what we have learned already is that CRB1 interactions are linked to cytoskeletal organization of photoreceptors and outer segment formation. We have actually identified a core interactome, and it's all about cytoskeletal reorganization. So the hypothesis is that mutations in CRB1 lead to perturbations in the actin cytoskeleton, leading to photoreceptor stress and cell death. And what does that mean in reality? And this is actually work that others have been done, Vladimir Arshavsky mainly, where he has shown that the actin cytoskeleton and its role in photoreceptors is to shape photoreceptor morphogenesis and organize outer segment formation. And that's very clear. And it happens where CRB is active, here in the kind of base of the outer segment where the outer limiting membrane is situated. And so to sum up, we are in the midst of defining what CRB1 is doing in retinal organoids and in the intact retina. And we have defined an interactome. We are now looking on how a potential genetic modifier will interact with this core function of CRB1. And it's a very interdisciplinary work, which we in the future want to combine and complement with Pete Quinn's work at Columbia and tightly work together on this. Thanks a lot.
Thanks, Marius, as always. This is just, a word of wisdom to being careful and figure out indeed the whole system before you start to treat the part. Nothing is worse than to treat and then it regresses and the damage comes 3 years later or 5 years later, and people see paradise and then they're jerked to go back to hell or whatever. This is just let's just go at the reasonable pace. Führer, I hope you help us to be proactive, and Marius to hold us back a little bit with some precaution. So there we go. And now we hear from Megan de Benedictis from the Cleveland Clinic. She's one of the genetic counselors in the field, and she will tell us about work and the importance of family support groups. Megan.
Hi, Irene, and thank you so much for the introduction. Hi, everyone. I don't have a presentation. I just wanted to very quickly talk. I know that we're running a little bit over, so I will just try to highlight how important it is that patients, families, researchers, clinicians, that we all really work together to help accelerate research efforts. And I think we all have a really critical role to play. But first, I want to recognize that so many patients and families spend years in this diagnostic odyssey, and they're wishing and they're hoping for someone to simply see them not just as a patient, but as a child, a sibling, a friend, a grandchild, see them as someone who needs help, and most importantly, see them as someone who is in need of hope. And hope is why we are all here today. One of the most critical steps of this whole diagnostic odyssey is having genetic testing done. So this is the best way to obtain a precise diagnosis, and we should all be pushing for and pursuing having that genetic testing done. Having a precise genetic diagnosis allows for the identification of patients who may qualify for and benefit from the various therapeutic interventions that we've heard so much about so far today. The second most critical step is being referred or referring patients to advocacy groups and research organizations such as CRBF. Having a centralized home and resource for patients and families that not only offers emotional and adaptive support, but it also creates a referral source of patients for clinical trials. We heard earlier today the word feasibility. We heard that mentioned a number of times in Fyodor's talk. And this is not just referring to the feasibility of a drug as a potential treatment, but for those of you who are unfamiliar with the process of clinical trials, when researchers, when companies, when clinicians are trying to determine the who, what, when, where, and how of a clinical trial, one of the first things that they have to consider is, how is this trial going to be feasible? And there are so many factors that play a role in feasibility. And one of those factors is, do we have the patients? Do we have patients who are eligible to make this trial feasible? And having patients enrolled in a registry that Kristin may mention earlier, I think she mentioned it when she first introduced herself, having a centralized home for patients gives us an inkling of how feasible is this trial going to be. And this is where CRBF and other patient advocacy groups become really, really critical, is having that centralized home and that data repository. CRBF has taken so many steps to spread awareness. They have presented at numerous ophthalmic education symposiums. They present at scientific webinars. They've presented to us as genetic counselors and other clinicians. This is truly such a unique foundation in that it is targeting one specific gene, the CRB1 gene. They've really stepped out of the shadows from all of the other inherited retinal disease genes. But they've also simultaneously served as a model and are driving research progress for all forms of genetic blindness. As CRBF is taking steps, so can all of you. There are so many ways to become active with the foundation, to fundraise, to engage with others, share in this magnificent mission, as Kristin was referring to it this morning. This is all synergistic. The patients, the ophthalmologists, the genetic counselors, the researchers, the advisory councils, the donors, we are all working together to achieve a common goal, the goal of being able to see and to be seen. And with that, I will turn it over to Kristin. Thank you.
Thank you, Megan. So before we hit the Q&A time, I wanted to thank all of our speakers here. And I wanted to point out that, you know, for, I'm sorry, I'm trying to do the technology while I'm talking. As you're hearing, I know some of this, you know, we all are coming in from different listening skills here, from different work that we do. But what I'm certain that all of you can hear is that, as I was saying, that we are funding the finish lines. We have multiple finish lines that we're looking at. This disease is complicated. So we are looking at multiple finish lines, and we've got sprints and we've got marathons. We've got halting progression, reversing the degeneration, prevention of degeneration to begin with. So there are multiple races that we're in, but we are funding finish lines now. And our call to action for all of you, as you just heard Megan say, that there are things that every person listening can do. You can get into the registry. Ophthalmologists and retina specialists can direct patients to us. And, the grassroots fundraising got us here. The 1.4+ million over the past 10 years got all this basic science and network building and finding the cohorts around the world that have all come together now to collaborate. And we still need that grassroots fundraising. We also need the connections to the bigger funds that will get us to those finish lines. Like we said for the title of this, accelerating it. For those of you that might be thinking, Fedor said 6 million. There's also opportunities to put some teams in place at 2.5 million. There are costs to keep all of this running that are up there as well. But when you think about this, we have patient leaders with cohorts of patients that have been talking from Spain, the United Kingdom, the United States, Portugal. We're reaching out now to the group in Puerto Rico. I mean, this is a global effort. And when everyone is collaborating in a global effort, it's no longer just one person trying to make a difference or one small team or one little group of patients. All of us pulling together are how this is going to get to the multiple finish lines. So what I want you all to come away with is the possibility is here. We are looking, our eye is on all of the finish lines, and we just need you to get us there. So with that, because we are running a little over, I'm gonna get to I'm gonna turn it back over to Irene, and we're gonna host a little bit of the Q&A, if I'm right.
Sure. Is it okay? Actually, Marius walked out on us, but I may ask the same question to Fiona and Pete. Do you think we can restore the cytoskeleton? That seems like the big challenge in this disease. The retina is basically finished, let's say, at age 5, or there's some maturation that's going on for the next couple of years, but it's already to a large degree finished at birth. And how are we going to reverse all of this if we want it? Is our goal to stop degenerative changes, or is our goal to get back to obviously our goal is to get back to 2020, but is there any reason to hope for that? Does anybody want to take that honor? Is it asking? Fiona, you kind of seem ready to give me an answer.
A general one, but a relevant one.
Good.
The Drug Administration and the EMA in Europe understand the concept of what's called a target product profile. It's probably the single most important acronym for all of you to know. What is that? It's a piece of paper on which it says, this is the disease, CRB1 LCA. This is the therapy, let's say CRISPR prime editing. The minimum efficacy profile is it will do the following. Restore X% vision in Y% of subjects who are older than Z. The optimal therapy, the optimum product profile is cure everyone. So the agency understands that you have to start small, and they will support a first-in-human intervention if you have animal efficacy data that support your minimum target product profile. Specific, I'll give you a specific. Sickle cell disease, you need thalassemia, you need 2 transfusions a month. You could go to the clinic by saying, we'll reduce the transfusion requirements by 50% in 50% of the subjects. The optimal TPP is 100% of the subjects will be 100% transfusion independent. So Irina, the answer to your question, broadly speaking, is you don't have to settle down to a specific answer. You simply need to have animal efficacy data that support a minimum efficacy profile in your target product profile. With respect to prevent versus treat, the entire in vivo editing space is fighting with this. Rett syndrome, Dravet, on and on and on and on and on. The animal data should really be guided. Can you take a mouse with established disease and repair the mutation and see what happens? If you have any evidence for restoration of anything, you can treat an established disease. If the answer is no, there are paths to be able to treat early stage symptomatic individuals, including in the pediatric population, if the agency understands the risk benefit. So it's not a zero-sum game. But the good news is relatively straightforward experiments will guide you to which mode do you want to do. Typically what people do is dose older folks just to see a safety signature. And if it's safe, there is some evidence of on-target engagement, but only modest efficacy, if any. Then the agency will say, fine, you can now dose early-stage symptomatic pediatric subjects. So I will shut up, except to say, as I mentioned in my 4-minute rant, all the bits and pieces are solved for you by other gene therapy and gene editing programs. There are no unknown unknowns, as Don Rumsfeld said. There are only known unknowns, and you just have to focus on them.
Yeah, and just to add to that, so we know from Luxturna now that it's not a cure in terms of it prevents, there's some degeneration afterwards, and there's some complications with this gene therapy. And that kind of goes back to what Marius was saying, is that maybe what we need to start doing is have these kind of combination therapies. So prime editing might fix the mutation, and then that will stop part of the degenerative process, but actually that degeneration is already ongoing from birth. And then maybe we need to think of some other type of metabolomic type therapies to also introduce at the same time to kind of stop this ongoing process. So I think in the future, it will be more of a kind of a broad approach that we need to facilitate.
Basically, do we need large animal? That was so productive for the Luxturna treatments, was the dog. I think those times are gone.
Irene, if there is no animal model, then the agency will not make it. I'm sorry to interrupt. If you have a dog, use it. But if you don't, the FDA will never say, make a dog and then you can treat a human being.
Exactly. We don't have a dog.
You have to do, and that's the expensive part, Kristin, is you have to do biodistribution in non-human primates. And that's expensive. That's $1.5 million. You need professionals to do this. You cannot go to the clinic without it. But that you just fundraise for that and do it. But Irene, I'm sorry to cut you off. I know our time is short. You do not need a large animal model if one doesn't exist.
That's exactly it. The mouse is a bad model for CRB1. So it doesn't express it in the same form that it expresses other things. It depends very much on the type of mouse you have. The Jackson Lab mouse is doing better than the NIH mouse. And so there are issues with the mouse, and it would be nice of maybe trying rat or trying something else and see what happens. But I think we'll leave that up for another discussion, and we'll get there. I think one of the things, and there I get back to Megan, what's happening is that people all over the world and the various groups are getting together to, to pool their force and their energy for the clinical trials. And I think that is happening at a very nice way. I think what's always a big issue in these parent support groups is that there are issues, which mutation do you treat first? How do you go about it? My kid I spent more money on it, and my kid is more important than yours, or stuff of that sort. And I hope we can avoid it in our organization and bypass that and do just go with feasibility and move forward with one gene and one mutation at a time. So there is competition, whatever competition means. Maybe there are different people running at the same time, from the Netherlands to France to different countries. And I think the more the better, and in the end we will know which one works the best and where is the success, and we'll just plod along until we get there, and until we have it right. So that's basically, the mission I have, and we'll go slowly and peacefully and move forward with it. As Kristin said earlier, do help and support this, and I never make that plea, but I think today and now is the time to make that plea, give to the CRM1 Foundation to help us move forward and fulfill everybody's wishes and be sort of proactive, given the support we have gotten over the years, and starting from very small beginnings and bake sales and small lunches and bicycle rides around the pond to going to bigger ventures in order to get to the finish line, as Kristin pointed out. And so I really thank you. Fyodor, you have another, I know the Russian word, you know, I should get it straight. Fyodor Dostoevsky is pretty fancy. So your name is known, so you go right ahead.
30 seconds. As far as the 6 million, folks, my job is to be honest. This is what it's going to cost end to end, but believe me, every little bit helps. And this is for the following reason. Success begets success. The more we know as we transition through the path, which is a 4-year path, the more clarity there will be on what exactly to do, what to deliver, how to edit, what the mouse model is, what kind of monkey model. And so the community right now, as I look at the entire sort of team CRB1 LCA that Kristin has gathered on the call, all the bits and pieces are there. So moving them along to sort of the next year and a half of where we have more definitive data on mode of editing, mode of delivery, animal model, target product profile, will be really enabling for the next stage. So I don't want to demotivate anyone with this price tag. It's not how this works. Getting to the next stage is essential, and that's not that much.
Okay. Kristin?
Well, thank you all for taking the time today to present this information, to ignite this mega campaign that we are looking to do this $3 million in 6 months. By the time we get to the anniversary of this foundation, my dogs are cheering me on there in the background, that we get to, in the next 6 months, get that initial 3 million to propel these finish lines that we're looking at and really ramp up and take advantage of these incredible breakthroughs that are happening in this space and continue to be a leader in the field. And like several of us have mentioned today, we did bake sales and bike rides to get us to this point. We still need the bake sales and bike rides from all of the CRB1 families, supporters, and friends. At the same time, and I dropped an email in the chat there, curingretinalblindness@gmail.com. Connections you have to funding. If you are sitting, I'm seeing some folks in our participant list that are potential collaborators for funding, partnerships, direct us to the collaborations. I think in some other language, CRB1 means collaboration, because that is how we got this far. This is not a 1-person, this is not a 5-person team, this is not a 10-person mission. This has been hundreds, thousands of people have been involved in this, and that's what we need to continue moving forward. So thank you to all of you that took the time today, that take time every day to move this mission forward. Please reach out to us at curingretinalblindness@gmail.com, and all of the CRB1 families, please be sure you go to crb1.org. Everybody can go there and donate today, but families, please go there and see the information on the registry and getting in connection with us so that you're not on this trip alone ever. Thank you so much, everybody.
Thank you.